Hi, my name is Lauren Proct, and I am a member of the group with Caitlin Dressendorfer, Caroline Martin, and Cubine Lee, and we are presenting on the topic of signal transduction, and more specifically going into how signal transduction is important in cancer. Now, what is signal transduction? Well, in Chapter 15 of our textbook, Molecular Cell Biology, signal transduction is defined as the process of converting extracellular signals into intracellular responses and all of the steps that are involved in that process. So you can see in this diagram here, I'll go into more detail on the specific steps later, but the signaling cell releases molecules that bind to a receptor on the target cell and induce intracellular responses. Now, there are 4 different methods that this signaling can take place through. There is endocrine signaling, paracrine signaling, autocrine signaling, and plasma membrane activated protein signaling. Now, endocrine and paracrine are both distance signaling. However, endocrine is a far bigger distance signaling than paracrine. Autocrine is within the same cell, and plasma membrane activated protein signaling takes place between 2 different cells that are attached through plasma membrane proteins. So here on this slide, I have different depictions of these types of signaling. So in the top left, I have endocrine signaling, where you can see that the signaling cell releases the signaling molecules into the blood vessel, and these molecules can then travel what is potentially a very long distance to bind to the receptors on these target cells. With paracrine signaling, which is down in the bottom left, the secretory cell is releasing molecules that are going to bind to receptors on an adjacent target cell. So they are not connected, but they are close together, so that even though these molecules are going into the extracellular space, they don't have very far to travel. With autocrine signaling, here in the top right, the signaling molecules are released from the cell, and then they go through and bind to receptors on that same cell. So they're still going into the extracellular space, but they are not binding to a separate cell. And then in the bottom right, with our membrane-attached protein signaling, the signaling cell and the target cell are both attached through these membrane proteins. And so the signaling cell activates the receptor proteins in the adjacent target cell to promote these intracellular responses. Now, going back to that original diagram that was on my first slide, I'm going to go into a bit more detail on the specific process of signal transduction. And of course, this can vary between different types of cells and different types of signaling, but it's generally the same idea. So in step 1, you can see that the signaling molecules are actually being synthesized in the signaling cell. These signaling molecules are then released into the extracellular space via exocytosis, as is depicted here in step 2. Now, step 3 is the actual traveling of the molecules to the target cell. And of course, going back to our different types of signaling, with endocrine signaling, this step 3 would be going through a blood vessel for a potentially very long distance, whereas with paracrine and autocrine signaling, they would only be going through the extracellular space for a very short distance before these molecules either bind to the receptor or are degraded. Which, of course, the binding to the receptors is depicted here in step 4. And these receptors are specific to the type of signaling molecule that is being released by the cell. So just because the signaling cell releases a chemical molecule does not mean that that chemical molecule is going to bind to any and every cell surface receptor. It will only bind to the receptor that is specific for that signaling molecule. Now, once these molecules bind to the receptor protein, then this binding triggers a response within the protein to activate the intracellular pathways as shown here in step 5. I'm going to go to our next slide. So once these intracellular response pathways are activated, there can be a couple of different pathways that actually get activated within the cell. If a pathway in this group labeled 6A were to be activated, these would lead to short-term intracellular changes in things like metabolism, function, or the movement of the cell. So once this pathway is activated, these changes will be induced, and then once the pathway is no longer activated, those changes will no longer be relevant. So that's why they are short-term changes. However, for a pathway in 6B, these are going to be long-term changes in something like gene expression or the development of the cell. So once this pathway is activated, that change doesn't get reversed, and it's going to be there for a while, unlike in 6A. Now, just because a signaling molecule binds to a receptor does not mean that these intracellular pathways are going to be activated. In step 7, the molecule binds to the receptor, but then the signal molecule within the cell to trigger those pathways is actually terminated by something else within the cell. In step 8, the signaling molecule outside of the cell is removed from the receptor or is terminated so that the intracellular process never even gets activated. So these are two different methods of terminating these signaling pathways before a response within the cell can be triggered. So now we're going to go into a bit more detail about how signal transduction can specifically be incorporated into cancer pathways.
Hello, my name is Kibin Lee, and I want to talk about one area of signal transduction that is very important in cancer cell for both cancer cell growth and cancer cell survival, and that is the receptor tyrosine kinase. And the one I'll be specifically talking about will be the AXL receptor tyrosine kinase. So the AXL receptor tyrosine kinase is a part of the TAM family, and it essentially has two Ig domains and two fibronectin type 3 domains on the extracellular domain or extracellular region. And it also has a kinase domain in the intracellular region, which I'll later talk about, which is associated with regulating the downstream signaling pathway. And like other receptor tyrosine kinases, there's an extracellular region, a transmembrane region, and an intracellular region. Now, although this is present in normal cells, it is also beneficial for cancer cells as well, as it can lead to potential oncogenesis, cell invasion, and drug resistance. And because these AXL receptor tyrosine kinases are abundant in a large variety of cells in the human body, they're found also in a variety of different cancer diseases, including breast cancer, renal cell carcinoma, prostate cancer, lung cancer, and so on and so forth. Now, the AXL and cancer, there are a lot of connections between how AXL contributes to cancer cell growth, survival, and resistance against our ways of stopping it. But AXL is essentially first activated by a ligand called Gas6. And this Gas6 has a high affinity for AXL as compared to other TAM family members, and it triggers downstream signaling by homodimerizing the AXL and allowing the kinases to interact with one another and phosphorylate each other via transphosphorylation. And this allows for effector molecules and adapter proteins with the SH2 or PTP domain to bind to it, and then therefore continuously go down a downstream signaling pathway until it reaches its necessary region of signaling. Now, this AXL receptor tyrosine kinase is generally present in a large amount of normal cells and tissues. Some examples being the platelets, the heart, the liver, the lungs, so on and so forth. And the difference between a normal cell and a cancer cell is that in adults and normal cells, there are generally a small amount of AXL present in each cell, but in a malignant cell, there is a large overexpression of AXLs present. And this can also lead to ligand independence, as the AXLs will generally aggregate with one another and dimerize even without the presence of ligands because of its large quantity. And it will thus trigger a large amount of these pathways even without ligand control. Not only that, but XL doesn't just bind to each other through homodimerization. It can also undergo heterodimerization, in which it binds to other receptor tyrosine kinases of the non-TAM family. Now, I'm going to continue on and explain the different ways that AXL actually contributes to cancer cell growth or evasion of any inhibitors. And that is the first one being cancer cell proliferation. Now, because the signaling pathway undergoes and goes into NF-kappa B, PI3K, and RAS-RAF, these are all signaling pathways that lead to cell growth and survival. And one way that it is actually enhanced by AXL is by apoptotic suppression or apoptosis suppression. And this is due to the regulation of translocation by AXL, and that allows for non-apoptotic proteins or anti-apoptotic proteins to survive in BCL, XL, and BCL2 to be produced much more. It is known, however, that stopping AXL, such as by using a short hairpin RNA to knock down the AXL gene, is known to restore the pro-apoptotic proteins that are needed in the cell so that the potential cancerous cell can undergo apoptosis. Now, the next one is cell migration, and cell migration occurs because of the AXL, because there are phenotypic changes that occur within the cell, such as an increase in GTP binding proteins RONRAC and the further development of filopodium. AXL regulates the epithelial-mesenchymal transition and thus allows the cell to change from the epithelial cell that has a cell-to-cell adhesion and is generally not very migratory and will not give much metastatic effects, and change it to a mesenchymal cell, which is a far more migratory and invasive cell, and with thus be moving around more. And this phenotype is maintained by the AXL presence. And if there's a larger amount of AXL present, then this phenotype is maintained more. And the final one is drug resistance. And this is essentially when there's an increase in AXL expression when a molecular targeted therapy is present in the cancer cell. And this can lead to more dimerizations of AXL, especially heterodimerizations with non-TAM receptor tyrosine kinases, and this can have an effect on drug resistance. Now, an example of that is the HER2 in the breast cancer cell. There is a drug for it called lapatinib that is able to inhibit HER2 and ultimately prevent breast cancer. However, AXL is able to bind to HER2 and prevent the inhibitory effects of lapatinib. It was shown, however, that once AXL is removed or inhibited, then HER2 would once again be susceptible to lapatinib, and therefore the inhibition will be present only when AXL is inhibited. So because AXL has such a large influence in cancer development and cancer survivability, there is recent focus on AXL inhibitory drugs, and there are future drug developments that are working underway that are under clinical investigation. And the focus and the hope is that this would potentially stop or prevent, at least for the time being, like the growth or spread of cancer by AXL influence. There are current AXL selective inhibitors that are created, such as SGI 7079, as you see here. And these are all ATP competitive inhibitors, which essentially means that they bind to the ATP binding region of the kinase domain and thus prevent ATP from binding and giving phosphates so that the kinase can phosphorylate regions that are necessary for the recruitment of other effector molecules or adapter proteins with the SH2 or PTB domain to further pass on downstream signaling pathway to necessary regions of the cell. Thank you.
Hi, my name is Caitlin, and I'll be going over the next article so that we can delve deeper into signal transduction and cancer. In the journal article Frontiers in Oncology 2020, it explores the IL-17 signaling pathway and how estrogen receptors interact to downregulate this. Now, this all sounds pretty confusing, but we're going to go over some background information so that the experiment makes a little more sense, and then we'll go over the actual experiment, and finally, we're going to go over some implementations or applications so that going over this article makes sense and how it can be beneficial to the real world. So first, we'll go over a brief background so that this experiment makes more sense. To begin with, from the cancer.org, from the American Cancer Society that I retrieved on November 4th of 2020, this source gave some examples of what hormone receptor positive and hormone receptor negative really means. So hormone receptor positive means that there's a presence of hormone receptors. Likewise, hormone receptor negative means that there is an absence of hormone receptors. For example, we can look at the estrogen receptors in breast cancer. So if you have estrogen receptor positive, this means that you have estrogen receptors present. And then in contrast, estrogen receptor negative means that there's an absence of these estrogen receptors. Now, what does this really mean though in terms of breast cancer? Well, if you think about it, for patients with estrogen receptor positive, this means that we can try to treat them with hormonal therapy and try to alleviate some of the breast cancer symptoms. But if we have a estrogen receptor negative patient, that means that we can't use this hormonal therapy to try to alleviate their symptoms. And the case for this is because if we don't have a receptor present, then putting more estrogen into the system is not going to solve any of the issues, since there's no receptor to bind this hormone that we're trying to give them. But in estrogen receptive positive patients, giving this hormonal therapy could just help them alleviate a lot of the symptoms because estrogen plays a key role in signal transduction pathways in breast cancer. For this, we can also kind of look into the IL-17, and this IL-17 family is a cytokine receptors, as we discussed briefly in lectures. Cytokine receptors are attached to the membranes, and from there we can learn more about these interactions of the estrogen receptors and the IL-17 and how we can figure what happens. So, secondly, we can talk actually about the experiment. So the researchers who wanted to look at the presence and absence of estrogen receptors and their effects on the downregulation of the IL-17 signal transduction pathway, they wanted to look at how this presence or absence would either increase or decrease this immune environment. Because the IL-17 pathway, a main part or product of this pathway is the PD-1 and PD-L1 protein, which is important in cell apoptosis and regulating that immune response in breast cancer. So, for example, if these proteins are present, then that means that the immune response in the breast will actually occur. And so what the researchers actually discovered was that the presence of ER, or estrogen receptors, actually downregulates this IL-17 pathway. Or in other words, when there's a higher level or higher concentration of estrogen receptors, then there is a lower amount of IL-17A, which was the main product or the main secondary messenger in producing that PD-1 and PD-L1 protein. And so this means that it also makes sure that this immune response isn't going into overdrive and that the body isn't trying to just attack itself. For this reason, we can see that it's important to have every single step mapped out in a pathway and to try to understand it better. This goes into, lastly, our implementations and applications. If we better understand a pathway like the IL-17 pathway, then we can better understand how we can attack or how we can treat this particular disease. With the estrogen receptor positive, we also saw an increase in IL-17E, which is an inhibitor in the signal transduction pathway. Knowing this information, we can see that if we have a higher amount of estrogen receptors, that the immune response is going to be blocked. But if we have no receptors present, as in the estrogen receptor negative, then there'll actually be this lack or this overdrive of immune response due to the fact that we don't have this estrogen receptor to downregulate the signal transduction pathway. And this patient will experience their own T cells attacking other healthy cells and allowing for the cancer to metastasize. With this, we can think about and understand that signal transduction plays an important role in all of cancer. And then finally, we'll leave you with some remarks to try to tie up all of our presentation.
In conclusion, signal transduction is a very interesting and complex process in molecular cell biology, and its understanding is necessary to study several cellular processes, such as the development and metastasis of cancer. As discussed, signal transduction describes the mechanism where the responding cell receives signals from another signaling cell or from the environment. The signal, such as a ligand, binds to the cell surface receptor located on the plasma membrane of the responding cell, which produces specific biological events to occur. The binding of this signal to the receptor located on the responding cell is greatly determined by the cell's sensitivity, which depends on the receptor's affinity for the signal. The signal transduction pathway and its role in cancer development was what we focused on, and especially the AXL and IL-17 pathways. AXL specifically is a receptor tyrosine kinase and has multiple downstream pathways that, if affected, can lead to several different cancers. On the other hand, IL-17 has shown how the ER presence can downregulate IL-17 and lead specifically to breast cancer. The disruption of the signal transduction pathway that cancer induces results in several of the cancer survival mechanisms, such as metastasis. The understanding of cancer development in general is a complex topic due to normal cellular processes suddenly going rogue. It is evident within the signal transduction pathway how normal processes such as the RTK pathway can show overexpression leading to cancer production. In addition, the complexity of the signal transduction pathway leads to limitations to several cancer treatments. The limitations to develop effective cancer treatments are due to several elements such as multiple kinases and proteins involved in the signal transduction pathway. For instance, by targeting a specific step in the signal transduction pathway to inhibit, several other kinases and pathways can be inhibited, leading to an adverse effect. To address the complexity of cancer and develop effective treatments for patients, scientists are continuing to work hard to understand the signaling transduction pathway. As evident in the article focusing on the AXL receptor tyrosine kinase in cancer development, AXL inhibitors, a treatment, appear to be promising. However, further research needs to be done to minimize the adverse effects that could be caused by the inhibition of the AXL receptor in the signal transduction pathway, and to also minimize drug resistance, which was evident in their clinical trials for this specific AXL inhibitor. Furthermore, other studies are being done to study the signal transduction pathway of IL-17, specifically in breast cancer. By studying the signal transduction mechanisms of cancer production, scientists are better able to hypothesize and test what treatments would be most effective for the cancer. Furthermore, directed therapy and specific understanding of the signal transduction mechanisms related to specific types of cancer, such as breast cancer, may help develop specialized treatments for specific cancers, such as breast cancer. Ultimately, to produce effective cancer treatments, further research needs to be done on signaling transduction pathways and their involvement in cancer development. Although the future of cancer research is unforeseen, the signaling transduction pathway and its role in cancer development
brings promise to provide effective cancer treatments to patients. And then here's our works cited pages as well. This is for the AXL receptor tyrosine kinase articles that we used. And then here are the citations for the breast cancer and the IL-17 signaling transduction pathway. Thank you.